divisions. The pars distalis is the largest division. That's where most of the endocrine organs, endocrine cells are. We have the pars intermedius, which is a strip between the adeno and neurohypophysis. It is glandular in origin, but it's not very dense as far as granularity usually. And then we have the pars tuberalis, which is the part of the adenohypophysis where it begins about midway down through the infundibular stalk. You'll start to see it in there. And the top pars tuberalis, again, it's got glandular tissue in it, but the most dense part, of course, is the distalis. Now, the main thing that is concerning about the pars intermedius, when we get into talking about tumors that secrete too much ACTH, if they're present in that area, they do not respond to some of our diagnostic tests like if they're in the distalis like they do. We'll talk about that when we get to it. Okay, you all seen this before, the feline pituitary. You can see the bigger portion at the top, that is the pars distalis. You can see the little glandular portion going around the nervosa, that is the intermedius, and this really doesn't have the tuberalis on it on this particular one. Now, as far as the adenohypophysis is concerned, it has the most wide range of secretory hormones than any other endocrine organ in the body. So it secretes the most. Okay? And it basically has different cells in there. They stain differently. They will stain either cytophilic, basophilic, or they won't take up the stain with an H&E. But the only way you can tell what they secrete is to do immunohistochemical staining for that particular hormone that you're interested in. Okay? Now, they are, as we said, secretory cells. They're in clumps around fenestrated capillaries. What's the purpose of having fenestrated capillaries? Right. Would it be, would you want to have to wait till that hormone trudges through that intact membrane to get into the blood, or would you rather have little holes that can go to the retina? And that's why we have the fenestrated capillary. Now, the access to circulation promotes the uptake of these substrates for hormone synthesis. In other words, you've got to have proteins and things like that to get in there, right? It also allows for very efficient release of the formed hormones into the circulatory system. Now, secretory activity of the cells is mainly controlled, as we learned earlier, through the hypothalamic factors, but there are also some ions that can affect it. There are some changes as far as vasal pressure and all this type of thing which can affect it. But mainly it is under the control of the hypothalamus. Remember, with the neurohypophyseal portal tract, basically the portal tract, what's portal mean?
Channel?
Portal, well, when we talk about anatomy and stuff.
Oh, liver?
Portal, no, not that type of thing. Portal means a capillary bed, veins between two capillary beds. So we have the capillary beds up in the, the hypothalamus, where all these stimulating or inhibiting hormones are released from the hypothalamus. They then go through these veins down to the adenohypophysis, and then we have another capillary bed that goes around those secretory cells so that they can respond. Okay, now what's a tract mean? When we say the hypophyseal tract, what's that mean? The tract is the nerve, that's the neurohypophysis. That's the axons coming down from those supraoptic and paraventricular nuclei up in the hypothalamus down to the neurohypophysis. So you need to know the difference between a tract and a portal system, so you don't get those two confused when you see it sometime in the near future on the exam question, okay?
Okay.
Can you repeat the tract?
The tract, when we talk about the tract, neurohypophyseal tract, we're talking about, remember the neurons up in the hypothalamus send their axons down to the neurohypophysis. And that tract is just those axons coming down. Okay. This again, you've seen this before, it's just showing you the diagram, showing you the portal system. You can see the arrows are pointing to that portal system right there. We have the capillary bed up in the hypothalamus, empties into those veins, and the veins then break into another capillary bed. And then that will release these hormones into the various cells so they can react to the stimulatory inhibitory hormones. And then they'll secrete back into there and go out into the systemic circulation. So that's basically how it's set up. And this little diagram here is in color showing you the same thing, also explaining basically what's going on. And notice that the neurons that are up in the hypothalamus, their axons don't go down into the infundibula. They stay up there and they synapse upon those blood vessels, those capillary beds up there. And that's how they release their inhibitory or stimulating hormones into there so they can affect the adenohypophysis. Okay. This is what the adenohypophysis looks like. You all remember that neuroepiphysis? This was kind of blob, it's just some fibers and neurons and stuff. So this is typical, glandular type appearance. Remember, they don't form the, you don't have ducts and stuff. Because endocrine doesn't have duct. Exocrine, where you have the ducts of these things. And these are endocrine organs. And you can see the capillary endothelium, little flat things in there. So it's very highly vascular. You can see cells aren't staining all the same color. And that's because these are the different cell types. Very rarely will you have one little lobule like that with just one cell type in it. You'll have some may have a predominance of one type, but then other cell types are also present in there. Okay, this is the infundibular of the pars tuberalis, and you can see it's just vessels coming down. Those are the vessels coming down. There are a few endocrine cells, as you can see up there in those clumps, but the tuberalis is mainly acts as a connection point between the hypothalamus and the pars tuberculosis mainly, but it's a connecting point for the adenohypophysis. And this is also showing the hypophyseal portal tract. If you see that there's this is neurohypophysis down here, this is adenohypophysis here, but these are all blood vessels coming down. Have a lot of that, capillary beds and stuff. And have gorged with blood, and that's taking the blood down into the pars distalis. This is the pars intermedius right here. And you can see the neurohypophysis is at the right up there. The adenohypophysis is over, it's a little off the screen, but it's down below. And so you see you just have a little bit of interspersion of some of the neural cells with the glandular cells, but it's mainly glandular in origin. Now, histologically, as we said, these cells can be named based upon their staining affinities. What's a chromophil mean? It likes stain, okay? And you have two types, acidophils, and guess what color they stain?
Red.
Red or pink, okay? And those are mainly associated with the cells that secrete growth hormone and also prolactin. Then we have the basophils. They usually secrete ACTH, TSH, FSH, and LH, but there is some intermingling. Now the chromophores, what's phobe mean?
Do not like them.
They don't like it, so they may stain pale pink or pale blue. Usually they'll stain a pale bluish color. They think that they're actually maybe not so much as a different cell type, or it might be just a different stage of activity of the cells. And they have, the ones that are weakly basophilic usually will secrete melanocyte-stimulating hormone and ACTH, but they have been shown to secrete all the other hormones. And that's why they think it might just be a different activity stage. If they're not as active, are they going to stain as dark or anything? So that's what they think they're associated with. So again, they may not be a separate cell type. It might just be a different activity. Okay? This is just showing you the cells. A are the chromophils rather, and B are the chromophores. And B will usually stain a very faint blue or very faint pink in there. Okay, this is another one. I don't know why they had to make A basophils. Why didn't they make B basophils and A acidophils, but they did. But you can see how these are staining, and you can see how they're interdispersed. So you're not just one lobe will be this, one's going to be that, so they're interdispersed in there. Okay, the chromophobes are some of the hardest ones to see because, as I say, they do have some color to them. So you say, well, they got some stain, but it's going to be much paler than the rest of the cells that are in there. Okay, here's just showing the different cell types again in the pars distalis, and notice that some lobes may have a few more one type, but most of them are interspersed throughout. This is showing you the more of an acinar type structure, but again, it's just showing you that around the outside surface we've got you can see this flat endothelium of the vessels in there. So the cells are very closely associated with these vessels, and they have to be. Okay. Now, when you use immunoperoxidase techniques, you can identify the cell based upon what hormone it secretes. Okay? So, what do you think gonadotropes secrete? What's gonadotropin?
FSH and LH?
In that instance, it's going to be FSH and LH. And it's about 10 to 15% of the total cell population in the adenohypophysis secretes gonadotropin. They're usually basophilic, but again, they can be chromophobic also. The staining is not really reliable when you doing that, that's why we use immunohistochemical staining. So FSH and LH can be used as markers. Both may be found in one cell. That is, they are bi or monohormonal cells. In other words, some of the gonadotropes will secrete both of those hormones. Some will secrete one or the other. Okay, so it's going to vary, and it may depend again upon what stage of the developmental cycle the animal is in, this type of thing. Also, there are some inhibitors which can do changes, just like inhibiting and all this stuff can change as far as how active these cells are. What do you think thyrotropes secrete?
Thyroid stimulating hormone.
Thyrotropin, the thyroid stimulating hormone. What causes them to secrete? Thyrotropin releasing hormone from the hypothalamus, okay? And they are about 10% of the total cell population. They are basophilic and they're periodic acid shift negative. We stain them like that. So we can also use specific staining for the TSH, or we can stain them with the PAS and get the negative stain. Figure out that's what they are. Somatotropes, basically these are rounded cells again with large granules. When you say angular cells, rounded cells, it's not always easy to tell on a histosection, especially if your sectioning wasn't too great and everything. So that's why we use the staining techniques, not any morphology to say what they are. As far as somatotropes are concerned, they secrete somatotropin, which is also known as growth hormone. Now, thyrotropin and thyroid stimulating hormone are the same thing. Okay, those are synonymous. Somatotropin and growth hormone are the same thing. Those are synonymous. Okay. Now, the somatotropes are the majority of the cells there. They're greater than 50% of the total cells. Does that make sense? What's your body need to do? It needs to grow and maintain itself, right? And so they're one of the major cells there. Now, they're acidophilic and PAS negative, but again, we have to use the special staining to use them. Lactotropes or mammotropes, they're used synonymously. They're variable size cells and they secrete prolactin. So what's prolactin do? Causes milk letdown, this type of thing. Now remember, the prolactin inhibiting hormone was what was secreted by the hypothalamus. Okay? So if we break the connection between the hypothalamus and the pituitary, which hormone will still be produced? Prolactin. Everything else is going to be inhibited to do that. Okay?
Repeat that.
If we break the connection between the hypothalamus and the pituitary, we will still get prolactin secretion, but all those other hormones will stop. Okay? Now, as far as these cells, they are about 10 to 25% of the total cell population. They're acidophilic, PAS negative. They will increase in number and granularity with pregnancy. That makes sense, doesn't it? What's a pregnant animal going to have to do? It's going to have to lactate, isn't it? Okay? And they basically, due to the effects of estrogen on them and also dopamine antagonists, reserpine and chlorpromazine, which are released with higher levels during pregnancy and parturition, this type of thing. In males and non-pregnant females, the granules are quite pleomorphic, and many are degraded by the cell's lysosomal system because usually does the male going to need a whole lot of prolactin? And if the female's not pregnant, do they want to have a whole lot of prolactin? No, they won't have to be lactating when they don't have to, right? They have a characteristic electron microscopic picture of exocytosis with a lot of granular extrusion. That's how they excrete their hormones, is by this exocytosis going on. Okay, corticotropes, they secrete adrenocorticotropic hormone, just ACTH. Also, they secrete beta endorphin. What are endorphins?
Feel good.
Feel good things. Well, sometimes, but anyway, feel good things. And also, they're all derivatives of a big molecule they call Big Mama, and it is pro-opiomelanocortin, POMC. And where you cleave this molecule, you'll get the different hormones coming from it. And melanocyte stimulating hormones and other hormones can come off of this molecule. As far as corticotropes, they're about 15 to 20% of the total cell population. They are basophilic, or they can be chromophobic, and they're PAS carnate. The throat's going on me. Positive. Most difficult. These are some of the most difficult cells to identify in sections of the pituitary because they can change their staining variable staining in there. And also, you usually will have to use either electron microscopy or actually use the immunohistochemical staining procedures for deposit identification. Now, what would it be essential to use immunostaining to identify these things? Well, if you have a tumor, do you want to know what kind of tumor it is? The prognosis will vary depending on what type of tumor it is. Okay, melanotropes, they secrete melanocyte-stimulating hormone, or MSH, and also lipotropins, this type of thing. They're mainly in the cells which secrete them are mainly in the pars intermedius of most of your vertebrates. And there's a reason for this, that the pars intermedius is going to have a more direct communication with the hypothalamus. And when do you want to have an increase in melon
When
secretion?
you're out in the sun.
No, not melatonin, melanin.
Oh, shit.
To protect against sunlight, all that type of thing, right? So light's going to change, right? And so you remember your light detection by your pineal gland and also by your hypothalamus. And so they can have closer contact with these cells when they're closer to them. So that's one of the reasons they're there. They're very similar as far as your corticotropes, and they actually may be the same cell. Remember that molecule can also make melanocyte-stimulating hormones, so depending on what enzyme is present in them will determine how that molecule is broken down and what you're going to get out of them. MSH is from the same precursor molecule as we said as ACTH. Okay, now here's just showing you an example of some histochemical staining. This particular one was for your corticotropes, and you can see the brownish coloration. Depending upon what stain you're using will depend upon what color change you'll get. Okay, now there's another cell type called the folliculostellate cells, and these are non-granulated cells, so they're not secretory. They're in the pars distalis and are on the same side of the parenchymal basal lamina as the granulated cells. In other words, they're kind of a communication between the blood vessels and the secretory cells. They tend to have processes that go between the basal lamina and the cells, so they help to give it some, strengthen it, give it some arrangements, this type of thing. And they do aggregate to form small follicles in the, or potential follicles, as we say. And they do react to one of the same antibodies as glial cells. So they think this is an adaptation of your microglial cells. They just haven't completely proved that yet. They've been seen to modulate the response of some of the granulated cells to both the releasing and inhibiting factors that they get exposed to. And they can be considered to be called paracrine cells, and their contribution of growth factors and possible interleukins is a very current thing as far as research investigation. So they may actually be some adapted secretory cells that have also got more of your microglial type properties. And this is just showing those big clear cells with the nuclei in the center. This is clusters of these parafollicular cells. That's what these guys are right here. Okay. And again, the reason they're interested in them is that there may be a means, if we can stimulate their development or inhibit them, depending on what the situation is, we may be able to regulate neoplastic processes up there, or we may be able also to regulate deficiencies of certain hormones and things like that. Okay, now the hormones of the adenoepiphysis are both trophic, and what's trophic mean, or trophic? Stimulates another endocrine organ, and that's like thyroid stimulating hormone, corticotropic release, corticotropic hormone, your FSH, LH, which is your hormones, and your non-tropic hormones would be prolactin, melanocyte stimulating hormone, and also growth hormone. Okay. Now the cells of the adenoepiphysis are under, as we say, hormonal, because that what's the neuroepiphysis under the control of? Neurons. So the adenoepiphysis is under the control of hormones. Okay, so it doesn't have direct neuronal type activity associated with it. They're directly controlled by your releasing and inhibiting hormones from the hypothalamus, and they can also be indirectly controlled by feedback mechanisms. In other words, if my glucocorticoids are very high in the blood, do I want to be secreting a bunch of ACTH? So it will inhibit that secretion there. Okay, and it can either be acting there or at the hypothalamus or both when we're doing that. Okay. Now, basically, we look at the anterior pituitary hormones, they usually can be grouped in 3 different families. Okay? Now, when we have these 3 families, this depends upon what molecule they were developed from. Okay? We have the glycoprotein hormone family, which means it comes from a glycoprotein. We have the pro-opiomelanocortin family, or Big Mama, and we have the growth hormone or somatotropin-related hormone family. So if we look at this first one, the glycoprotein hormone family, this includes LH, FSH, and TSH. And all members of this family basically transduce their intracellular effects via the receptor G protein. And then the second messenger and the cyclase and this type of thing. In other words, these are peptide hormones. They are glycoproteins that, actually the TSH is not that big. There's some of the smaller glycoprotein hormones. But again, it's basically produced in their, so where are the receptors for glycoproteins used? Cell membrane surface. Now, what's going to be the site for TSH? Where's it going to go to stimulate? The thyroid, and thyroid follicular cells. Okay. Of course, LH and FSH are going to go to the various reproductive organs and have their sites there. Okay. Now, they basically again are from 25,000 to 30,000 size, which is not really that big for most hormones. Actually, they're smaller than albumin, so which one of the ways we get them out of the blood? They excrete through the kidney, excrete through the liver, and proteases break them down. Okay. Now, basically, they're the most chemically complex family of peptide hormones. In other words, they have a lot of different structure as far as amino acid